So in Unit 5, what we'll be looking at is Mendelian genetics and also non-Mendelian genetics. And this is going to be the start to our study of heredity, or you can also call that genetics. When you're looking at the word genetics, it does automatically tell you what you're going to really be looking at, which is the gene. And just as a reminder for ourselves, a gene is going to be a section of DNA that codes for a trait. And when it codes for a trait, it'll actually be coding for a protein to eventually make our traits. So when you're studying heredity, what are we actually looking at? We're not just looking at genes, but we're also going to be looking at how traits are passed from parent to offspring. And usually we're going to be looking at through the generations. When we're talking about the passing of parents, this means that we'll actually, in a way, be looking at meiosis, or sexual reproduction, in more depth. Because sexual reproduction results in variation, we have to be able to make some predictions about what would a parent pass on to the offspring, what is the result of meiosis or fertilization. And many times we can actually identify certain repeatable patterns. And for some traits, they will follow specific rules or certain rules that help us to easier identify that type of trait. What type of traits am I talking about? We're thinking of sex-linked traits, autosomal dominant traits, or even autosomal recessive traits. When we're trying to study heredity, one way that we can do this is by looking at a family tree, which is also known in biology as a pedigree. There's some very simple patterns that we can automatically see, or some simple factors that we can identify. We can identify that there's 3 generations. Hopefully you remember that a circle is a female and a square is a male. When you have a colored in circle or square, that represents an affected person or a person that carries the trait. Sometimes for a carrier, you may have a carrier that is colored in halfway, and sometimes you won't have a carrier that's colored in at all, and it'll just depend. A lot of times we can also see that the trait may be recessive because not all of the children have the disease. Sometimes it can be dominant because the dominant allele is passed on and each generation is affected. But there are some problems with studying human genetics or human heredity. And because there are problems, we usually use other organisms or model organisms, such as the pea, like Mendel did, or the fruit fly, like hopefully we will do. When we're thinking about human heredity, really what we're looking at is inheritance. And so you have to ask yourself that question, what is being inherited? And hopefully you can answer that and say that that's DNA. DNA is being inherited through the sperm and through the egg. So problems in studying human heredity, what are some major issues or major factors that are holding us back? First thing is going to be our lifespan. The human lifespan is very long between the generations. And so this is a limiting factor for our studies of heredity. Another is the long gestational period. Takes us 9 months, approximately, to carry a baby. 9 months. And then we can't intentionally breed. Intentionally breed. So this is not a realistic idea. And what do I mean by intentionally breed? I mean, there's two parents and there's a recessive trait. We want to see if the parents are homozygous or heterozygous. Can we crossbreed them? Can we do a test cross? And really, this isn't even allowed. We're not allowed to have people breed just so we can see the result of their offspring. One other way that we will be able to look at heredity a little bit is through, and actually evolution later on, is through something called a phylogenetic tree. But right now we'll mainly focus on family trees and pedigrees, as well as with Punnett squares. So we can't talk about genetics without talking about Mendel. And it turns out that Mendel's experiments were very carefully designed. Some simple things to know is that he did use the pea plants because of several factors. One, they produce lots of offspring. And it was, two, easy to control the matings. He can actually cause the pollination to occur himself, so he could pollinate whichever flower he wanted with whichever other flower. This would be called cross-pollinating. And this is similar to sexual reproduction. Or he can self-pollinate the flowers, which would be kind of an example of asexual reproduction. But this would allow him to control which genes are passed to the offspring. Another good thing about Mendel and his studies is that he kept very meticulous data, something that we are working on. But he kept very meticulous notes, very meticulous data. And the other good thing about the pea plants is that the traits, each trait, had two contrasting forms, or you can even use the word that we looked at today, alleles. So every trait that he looked at, the height of the plant, the type of pea that was produced, round or wrinkled, yellow or green, there's only two contrasting forms of that allele, which made it a lot easier to study. So in the experiment, there's a couple things you want to look at. We just highlighting some vocabulary that we looked at earlier today. Crossing something is a mating or a Punnett square. Progeny is another word for offspring. I'm not going to read this to you because obviously you can read for yourselves. But a couple of other things I want us to point out and have you guys look at this on your own is your P generation is going to be your parent generation, or generation 0 if you think of that, or generation 1 actually. You can have a homozygous plants, homo meaning the same, for round or wrinkled. And this was also known as pure breeding, which is something that we talked about earlier. You can have your F1 generation. F stands for filial, which could be, in this case, your first round. So how many rounds are you going to have, or how many generations are you going to have? F, or you can even think of the F as fertilizations. How many fertilizations have you had? 1 or 2, depending on how many generations you're producing. And then last of all, here, when we look at the fraction, we'll be studying ratios, so how many out of the whole are we looking at? Again, I don't want to read this to you because you obviously know how to, you're on your own. But you want to think about, how did a trait skip a generation? If the traits of the parent plants do not blend, then what does that mean? And how would, I'll write that down, how did a trait skip a generation? And you should be able to give me this answer. But that's all going to be for this really quick intro, because we did go over most of this new vocabulary in class today.